Hello, my name is Jungsul Lee, working as a postdoctoral fellowship at Optical Bioimaging Center under Professor Choi on Bio and Brain Engineering Department of KAIST. Today, I will talk about our research how to find phenotype deterministic genes, which was recently published a scientific report at 2014 with the title Identification of Phenotype Deterministic Genes Using Systemic Analysis of Transcriptional Response. I will briefly talk motivation of this research. After that, method, result, and summary will be followed. Yuri Rajevnik published a seminal paper, Can a Biologist Fix a Radio at 22, 23, and 24. In the paper, the author used a metaphor for a biologist trying to fix a broken radio. A normal serviceman tries to find broken radio parts by checking resistance, impedance, or any other things with stubborn tools. Any broken parts can be found by checking response with given input signal. Let's apply research process in biology into this situation. Biologists try to find malfunctioning parts such as transistor or capacitor by looking at physical appearance of the parts, whether there is a scratch or something or not. Yuri emphasized on the importance of change of research paradigm in biology using this metaphor. My research followed similar logic of this paper. That is, if we want to find phenotype deterministic genes, then we should check relationship between input and output simultaneously instead of observing them independently. Let us apply the previous broken radio metaphor to phenotype deterministic gene identification problem. In this schematic diagram, if we check only response, then samples in blue circle would be considered as malfunctioning. However, samples in red circle would be turned out as abnormal when stimulation and response are considered simultaneously. That is, strong response of samples in blue circle stem from strong stimulation. Blue circle is normal. Samples in red circle show low response even with a strong stimulation. Therefore, we should consider strength of stimulation and response simultaneously to find malfunctioning components. In cells, relationship between activities of transcription factor TF and expression levels of their target genes can be considered as a stimulation and response or input-output relationship. If we compare the relationship on several groups with their own phenotypes, then we can determine which TF target gene pairs are related with the phenotypes. Here, genes in the same pathway with the TF will be considered as stimulator because the genes also can regulate expression level of target genes of the TF. This is an illustrative example. In figure A, there is no difference of expression level of genes that can be considered as stimulation. So do target genes in figure B. If we merge A and B into one plane like figure C, then it is evident that only group A has correlation between strength and stimulation x-axis and response y-axis. That is, it is likely that a different relationship can be found by checking two factors simultaneously even when the two factors have no difference. Figure D is made with real patient data. I compared the proportion of EGFR target genes with significant expression correlation with EGFR itself from tamoxifen-treated breast cancer patient samples. As you can see, there are more target genes with significant expressional correlation with EGFR in tamoxifen response patient samples than tamoxifen resistant patient samples. I will elaborate this basic concept and show how to use it to find phenotype deterministic genes. First, pathway genes should be defined. Activity of transcription factor is regulated by several other genes involved in the same pathway with the TF. Therefore, the genes can be considered as stimulation. I named the genes pathway gene. I used the pathways in NestLib and defined the pathway genes with the pathways in NestLib. Next, I defined the formula for the extent of regulation of the pathway gene using correlation between expression of the pathway gene and their target genes. Significance rate (SR) is defined as the quantity of how strength the pathway gene regulate well their target genes. Target genes of a pathway gene is defined as all of target genes of TF involved in the same pathway with the pathway genes, and then calculate correlation between expression level of the pathway gene and each of the target genes. A pathway gene is considered as being upright normal when the correlation is significant with the first discovery rate less than 0.005. For each pathway gene, the ratio of target genes with a significant expression correlation with the pathway genes is defined as a significance rate SR. That is, SR means the proportion of pathway genes, target genes that can be told regulated well by the pathway gene. For the two different phenotypic groups, significance score SS is defined as the ratio of SRs. After simple arithmetic conversion, the range of SS is restricted to 0 and 1 inclusive. In this work, I used the Spearman's rank order correlation coefficient, and target genes of TF are downloaded from MSigDB. Total 3 datasets were used for tamoxifen response and 1 for epinephrine response from GEO. In case of tamoxifen response, the 3 datasets were reclassified as total 8 datasets according to microarray platform because comparison of expression level with different platform is impossible. I used the raw expression value recorded in GEO after quantile normalization because Spearman's correlation is scale and shift invariant. This is the first result. I checked if the expression level itself is different between tamoxifen response and resistant groups. Only for 4 among 8 datasets, there were differentially expressed genes, DEG, with FDR less than 0.005 between the two groups. Moreover, the proportion of DEG is more or less than 5% of total available genes. Therefore, there is no gene expression difference between the two groups used in this research. It implies that method based on DEG would fail to find phenotype deterministic genes. On the other hand, as can be seen in figure B, quantification of stimulus response concept formulated as SR were found to be different between the two groups of all datasets. It means this method can be applied to cases where there is no expression difference between two different phenotype groups. Moreover, distribution of SR was maintained in different microarray platforms. When classified as tamoxifen response and resistant group, pattern of SR distributions were similar with different microarray platform, as you can see in this figure. There is a SR of tamoxifen response patient groups were larger than those of tamoxifen resistant patient groups. It means gene regulation is more stringently regulated in tamoxifen response patient than resistant patient. Only 6% of target genes of pathway genes were regulated in tamoxifen resistant patient, but about 60% of total target genes were found to regulated in tamoxifen response patients. Next, we checked consistency of SS because consistency has been a major issue of researches using microarray result. As can be seen in this figure, pattern of SS were maintained consistently even in research performed independently with different microarray platform. We selected the top 5 genes with the largest SS and then tried to in vitro validation. Before that, the top 5 genes were found expressed a similar level between tamoxifen response and resistant patient groups of all available datasets we used. For those 5 genes, we silenced the genes with siRNA and then tested if the cell death was influenced by tamoxifen treatment. That is, we suspect that The 5 genes were related with tamoxifen-induced cell death, so we silenced the genes and then checked tamoxifen-induced cell death by WST and FACS. We tested it in 2 breast cancer cell lines, MCF7 and MJMB231. It was found that JAK2 and SOCS2 silencing influenced tamoxifen-induced cell death compounded by WST and FACS, as in this figure. Next, we rechecked if target genes of JAK2 and SOCS2 have different expression correlation between the 2 patient groups. As in this figure, SOCS2 and JAK2 have more strong expressional correlation with their target genes in tamoxifen response patients than in resistant patients. Moreover, this pattern was consistently observed in all data sets. Even these data sets were performed independently with different microarray platform. In case of SOCS2, 2 data sets have different correlation value, but the statistical significance was observed only in tamoxifen response patient data. To sum up, identification of phenotype deterministic gene is a long history after the advent of high-throughput gene profiling. However, most of the methods utilize differentially expressed genes between 2 phenotypic groups. Therefore, the methods are inapplicable if there is no expressional difference as in drug response case. Moreover, using DEG showed inconsistent results in many researches. In this work, I proposed the significance score as the quantification of input, output, or stimulus response concept, and then used it to find tamoxifen response related gene. Especially, even all datasets I used to have few expression difference between drug resistant and response groups. The method successfully identified the phenotype deterministic gene. Moreover, this method can be applied to any case with different phenotypes other than drug responsiveness. This is the end of my talk. Thank you.